Hello everybody, welcome to the 3 small sub-lectures for our Synaptic Transmission lecture number 1. So in this first small video we are going to go over the basics of synaptic transmission, and the goals are going to be to learn about the basic characteristics of electrical and chemical synapses and to understand the main differences between those two types of synapses, and then to apply this knowledge of these differences to design experiments that would distinguish between them. My name is Maria Svetanovic. Below here is my email and also my address. I'm in volume building, so any questions or comments you have on any of the recordings you listen, please make sure to let me know. I would love to get your input. So synapse is a term coined by Sherrington to describe a site in between which neurons communicate with each other. It comes from the Greek word synapse, which literally translated would mean to fasten together. Here's a beautiful illustration of Ramon and Cajal in which he was showing us how the neurons are touching each other. At that point, the nature of this contact between the neurons was still not known. What we know now is the synapses in the nervous system coexist in two main configurations. One electrical, that you can see here on the left, and that can be distinguished with these on the electron microscopy with the darker both side, and then chemical, which is very easily distinguished by the presence of synaptic vesicles and then postsynaptic densities. Now these synapses are named according to the mechanisms that are involved in transmission, and there are very significant differences between two of those different types of synapses. Again, we're going to go through basic differences that exist between these types of synapses. At the basic level, as I said, electrical synapses allow the transmission of intracellular content from one neuron to another through the gap junctions, which are intracellular channels. And again, we are going to go a little bit more in detail soon how these gap channels are formed, but it allows the transmission of these ions that go through the gap junctions. So you're really literally connecting cytoplasms of one neuron with another. And then probability of transmission of any information is going to be always 1. That's really an important characteristic. On chemical synapses, we have a transmission that involves synaptic vesicles that are released in the extracellular space, and then they bind receptors on postsynaptic neurons. That means that there is no direct connection of one cytoplasm of presynaptic neuron with the postsynaptic neuron. But what we need here is an action potential that will lead to the release of synaptic vesicles that will then activate either ionotropic receptors, which will produce the current flows, or metabotropic receptors, which are not presenting in this image, that will be able to modulate this signaling in the postsynaptic neurons. This means that this probability of transmission information is always going to be less than 1, because again, the voltage change in the presynaptic terminal has to reach to the action potential for any information to be transmitted. And then this neurotransmitter that are transmitted need to bind certain number of receptors to again transmit information to the postsynaptic terminal. So what are the features of electrical synapses that I would like you to remember? They're rapid. There is no synaptic delay. So the moment the signal comes to the presynaptic neuron, presented here, this is giant motor synapse in the crayfish, which is where the electrical synaptic transmission was first demonstrated in 1957. So you have a really large presynaptic neuron and a small presynaptic. This is also important because, again, you will transmit more information if you go from a large into the smaller postsynaptic neurons. So the information goes directly because their cytoplasms are connected. This is shown here. If you monitor the voltage in the presynaptic neuron and postsynaptic, you will see that the change occurs instantaneously. They can be bidirectional because, again, these are gap channels. Information from one neuron can go from post to pre and from pre to post. Again, depending on the size of these neurons, how much will come will influence how much information is going to be transmitted in either way. So they can be bidirectional. There can be also unidirectional if there is a modification of these channels. And again, change the potential in the postsynaptic cell is directly related to the shape and the size of potential in the presynaptic cells, because again, it's a direct connection. And what I mean by that is illustrated a little bit better in the next. So in this next slide, again, we have the presynaptic neuron, you're monitoring voltage in presynaptic cells, and then you monitor the voltage in the postsynaptic cells. And you see you have the same shape. And then also the size is directly influenced. So larger the voltage would be in presynaptic, larger the pulse you would deliver to presynaptic cells, you will have larger voltage change, and you will have larger voltage change in the postsynaptic cells. As I mentioned, that we are going to go a little bit more in depth, not really too much, is how these synapses are made. We are learning much more that these synapses are actually quite interesting. They're composed of numerous gap junction channels. These gap junction channels are formed by two opposed hemi channels. So we have one on each membrane, on each neuronal membrane, and each of these connexons or opposed hemi channels are formed by 6 connexin subunits. They have pretty large diameter, around 1.2 nanometers, and that allows not only flow electric current that you want to go from one neuron to another, but you also can exchange the small metabolites and intracellular signaling molecules. You will learn a lot about calcium's role as an intracellular metabolite, but also CMP and IP3 that will then lead to more release of the calcium can go when these channels are open. For a long time, these synapses, electrical synapses, were not thought to be very plastic. Now we know more, and we know they allow for the plasticity, as the opening of these channels can be modulated by phosphorylation and also by insertion of the channels. So we know there's a constant insertion and removal of these channels that can lead to the plasticity of electrical synapses. In addition to this, most of these channels can actually close in response to things that are really damaging to neurons, such as lower pH or increased intracellular calcium. This is a really good measure to prevent the spreading of neurotoxicity from one neuron to another. On the other hand, we have chemical synapses, and I said they are characterized by this synaptic cleft, which is the space between pre- and postsynaptic neurons. The key features that you can easily see that is chemical synapse is the presence of synaptic vesicles, which are membrane-bound intracellular organelles that are filled with neurotransmitters, as well as a postsynaptic membrane. These release sites where neurotransmitters are going to be released are called active zones, and we are going to talk a little bit more of what constitutes the active zones. Now, there is a sequence of events during chemical transmission. So we have arrival of the presynaptic action potential. So as I said, it needs to reach certain threshold, the change in the voltage is presynaptic membrane, and that will lead to the opening of voltage gating calcium channels. This will allow for calcium to enter the presynaptic terminals, and this entering the calcium is going to lead to the opening or fusion of these duct vesicles, which are release ready. They're primed to release. When they fuse to the membrane, they're going to lead to release of neurotransmitters. These neurotransmitters are going to drift through this synaptic cleft and then bind to the receptors. As I said, they can be ionotropic or they can be metabotropic receptors, and that's going to lead to the opening of these, again, postsynaptic membrane channels, ionotropic channels, which is then going to lead to the change in the membrane of the postsynaptic neuron. Again, these steps are accounting for a significant delay. This is not instantaneous as we have in the electrical synapses when the ions can directly flow from the presynaptic to postsynaptic. Now we have all of these steps, and therefore after we have the arrival of the action potential peak in the presynaptic action potential, there's this delay before we can measure the voltage change in the postsynaptic membrane, and this is what is called synaptic delay. And how long the synaptic delay varies in the different brain regions, different neurons, and again depends on all of these steps. You know, how quickly the voltage gated channels open, how quickly the membranes fuse, how quickly the neurotransmitter can go through the postsynaptic channels. Very important thing for chemical synapses that distinguish them from electrical, and for a long time, as I said, it was thought electrical are not plastic, is the plasticity. And we know the plasticity is really the basic of learning. So here are examples of 5 different forms of plasticity, whether they're going to be postsynaptic or at the presynaptic terminals. So again, we can lead to the insertion of more postsynaptic receptors and perceptors that are going to increase how much signaling the postsynaptic neuron will see. You can also lead to the changes in presynaptic, let's say voltage-gated channels, so you will have more or faster calcium entrance, and that can lead to more release of neurotransmitters. But you can modulate again in many different ways the chemical synapses, and that is their key characteristic. For homework, I would like you to think based on what we have right now talked, that differences between electrical and chemical synapses, to think of different ways by which you can experimentally distinguish whether the connection between two neurons in the brain is going to be electrical and chemical. You can use different methods from immunohistochemistry to electrophysiology, whatever you want, but think of the ways you would be able, if I point you to the neurons in the brain, how would you be able to experimentally test whether they're connected through electrical or chemical synapses?